Good evening, ladies and gentlemen, and a warm welcome to the 33rd Annual Scientific Session 2020 of the Physiological Society of Sri Lanka. The Physiological Society of Sri Lanka is the academic body of the physiologists in Sri Lanka. Teachers in all medical faculties in Sri Lanka, as well as local and overseas medical professionals, are the members in this organization. A kind reminder to the audience, as we are going to start this session shortly, please be kind enough to mute yourself, to keep silent mode and turn off the video. To commence this session, please rise for the national anthem.
Sri Lanka Mata, Apa Sri Lanka, Namo, 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 Namo Mata, Sundara Sirivarini, Senaviratne Sobhamaya Lanka, Vanalaya Nekamal Palaturu Pirijaya Bhumiya Ramya, Apahata Sapasiri Seta Sadana, Jivanaye Mata, Piliganu Mena Apavakti Puja, Namo Namo Mata, Apa Sri Lanka, Namo 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 Mata, Ubave Apa Vidya, Ubamaya Apa Satya, Ubave Apa Shanti, Apa Hada Kula Bhakti, Uba Apa Aloke, Apa Ge Anuprane, Uba Apa Jivanane, Apa Muktiya Ubave, Nava Jivana Demine, Nitina Apa Kubudu Karan Mata, Jana Veera Vadavani Laragena Yanudana Jaya Bhumikara, Ekamavaka Daru Kalabavina, Yamu Yamu Vinopama, Devavada Samaveda Durada, Namo Namo Mata, Apa Sri Lanka, Namo 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 Namo
Now I call upon Professor Niranga Devanarayana, President, Physiological Society of Sri Lanka, to welcome the distinguished invitees and to deliver presidential address.
Good evening. Chief Guest Senior Professor Nilanthi De Silva, Vice Chancellor, University of Kelaniya, K. Senaviratne Memorial Orator, Vidyanidhi Professor Mangala Gunatilaka, members of the Executive Committee, past presidents, members of the PSSL, family members of the late Professor K. Senaviratne, ladies and gentlemen. I warmly welcome all of you to the 33rd Annual Scientific Sessions of the Physiological Society of Sri Lanka. Physiology in its broad sense aims for an integrated understanding of living mechanisms across a wide range of scales encompassing molecules, organelles, cells, tissues, and systems. It has been widely considered as the foundation of clinical medicine. Founded in 1987, the Physiological Society of Sri Lanka has been promoting physiology education and research in Sri Lanka over 30 years. The members of the PSSL has been helping to promote diverse research interests among the members, from molecular biology and genetics to clinical and applied physiology in different medical fields such as neuro, endocrine, reproductive, respiratory, and gastrointestinal physiology. Due to COVID-19 pandemic-related restrictions, this year the annual scientific sessions of PSSL is conducted on virtual platform. However, However, an excellent scientific program has been laid down for these sessions under the theme Physiology in Challenging Times, in addition to annual Kane, Senaviratne and Valentine Basnaik orations and the inaugural Carlo Fonseca Memorial Oration. I would like to express my deepest gratitude to the Executive Committee of the PSSL for their excellent work, which you are witnessing these 2 days. Finally, I appreciate the generous support from all of you and our sponsors, without whom this would not have been a success. Being appointed as the president of PSSL is one of the greatest achievements in my life. It is a great pleasure to deliver the presidential address for the year under the title, Quest to Find Solutions for Pediatric Functional Gastrointestinal Diseases. What are functional gastrointestinal diseases? Functional gastrointestinal diseases are defined as a variable combination of chronic recurrent gastrointestinal symptoms which cannot be explained by structural or biochemical abnormalities. Functional GI disorders in children and adolescents are categorized into 3 main groups. There are 10 main functional gastrointestinal disorders. I have been doing studies on most of these types, but most of my studies concentrate on functional abdominal pain. It is a worldwide problem in pediatric care, and its poor prevalence is approximately 13.5%. There are 4 main types of functional gastrointestinal diseases. They are functional dyspepsia, irritable bowel syndrome, abdominal migraine, and functional abdominal pain not otherwise specified. In 2001, it was Professor Asoka Disanayaka, former professor of physiology, University of Kelaniya, who introduced me to functional abdominal pain in children and encouraged me to conduct my postgraduate studies in this field. I'm extremely thankful to him for introducing me this interesting and exciting research field. Subsequently, under the great supervision of Senior Professor Janaka De Silva, I have conducted my postgraduate research activities for Doctor of Medicine degree, University of Kelaniya, on epidemiology and pathophysiology of this condition. I continued my research on abdominal pain in children and obtained my PhD from the University of Amsterdam under the guidance of Professor Mark Benninga, Professor of Pediatrics, University of Amsterdam. When I started my research on functional abdominal pain disorders in 2001, very little was known of its epidemiology, pathophysiology, and management, and there were many unanswered questions. During the last 2 decades, my research related to pediatric functional disorders focused on answering 4 main questions. They are, what is the functional abdominal pain burden in Sri Lanka and elsewhere in the world? What is the impact of these disorders on affected children? What causes functional abdominal pain and how can you manage this condition? What is the burden of functional abdominal pain disorders in Sri Lanka and elsewhere? Let's look at the epidemiology of functional abdominal pain in Sri Lanka. I have conducted several studies on epidemiology of functional abdominal pain in different parts of the country, including one island-wide study. The prevalence varies from 9.2 to 16.5%, which highlighted the heavy burden of this disease in Sri Lanka. These researchers also identified the commonest functional abdominal pain types in Sri Lankan children, which are irritable bowel syndrome and functional abdominal pain not otherwise specified. I have been collaborating with international researchers to look at the epidemiology of its epidemiology outside Sri Lanka. One systematic review conducted in Asian countries revealed a poor prevalence of 12.5%. I have also contributed to 2 studies which assessed prevalence of functional abdominal pain disorders in Nigeria and Curacao. And These help to understand the burden of functional abdominal pain in those countries. Second question I try to find solution is, what is the impact of functional abdominal pain disorders on affected children, their families, and the healthcare system? Research conducted in relation to quality of life in affected children have shown significantly impaired function in physical, emotional, social, and school functioning domains, and all, as well as reduction in total quality of life. Being a physiologist, the question that interested me most was what causes abdominal pain in children with this condition. The majority of my research concentrated on finding possible underlying pathophysiological mechanisms for this condition. Exact pathophysiology of functional abdominal pain is not very clear. Dysfunction of brain-gut axis is suggested as the main possible pathophysiological mechanism. Thorough understanding of pathophysiology is essential for effective management. Therefore, the management of functional abdominal pain is also not very robust and very ineffective. What is brain-gut axis? Brain has a direct effect on the gastrointestinal tract. For example, very thought of eating can increase gastric juices before food even get there. This connection goes in both ways. A troubled intestine can send signals to the brain, as well as troubled brain can send signal to the gut. Therefore, the person's stomach or intestinal distress can be the cause or product of anxiety, stress, and depression. That's because the brain and gastrointestinal system are intimately connected. They're so intimately connected, they should be viewed as one system. Many pathophysiological mechanisms have been described for functional abdominal pain. This figure we created for a review on functional abdominal pain in Nature Review of Gastroenterology highlighted the suggested pathophysiological mechanisms for this condition. My research so far have explored some of these pathophysiological mechanisms. They are early life events, psychological factors, adverse life events, gastrointestinal motility abnormalities, abnormalities in the brain-gut communication, gastrointestinal infections, and allergy and immune mechanisms. Emotional factors, including stress, have been considered as an important pathophysiological mechanism for functional abdominal pain disorders. In our studies, we have shown that children with functional abdominal pain are more likely to be exposed to stressful life events than children without this condition. The main school and family-related stressful life events associated with this condition are listed here. Adverse life events such as abuse have a significant impact on psychological and physical well-being and likely to have an impact on gastrointestinal functions. Previous study we conducted have highlighted the association between physical, emotional, and sexual abuse and different functional abdominal pain disorders. At any age, teenagers exposed to abuse are more likely to develop functional abdominal pain than those not exposed to such events. It is believed that individuals with certain personalities are more likely to develop functional disorders such as headache. Therefore, we evaluated the association between functional abdominal pain and personality types. In agreement with our hypothesis, children with abnormal personalities such as hostility and aggression, negative self-esteem, negative self-adequacy, emotional instability are more likely to develop functional abdominal pain. Psychological and emotional factors affect gastrointestinal tract. There are two main communication systems. autonomic nervous system, and hypothalamo-pituitary adrenal axis. Therefore, we have assessed the autonomic functions in children with functional abdominal pain. When compared with healthy children, the affected children did not have significant abnormalities in autonomic functions. However, unlike in healthy children, we observed an autonomic nervous system unresponsiveness in children with this disorder. Based on our findings, we managed to suggest a pathophysiological model to describe the impact of autonomic nervous system on development of functional abdominal pain disorders. This model is published as Automatic Stomach Model in World Journal of Gastroenterology in 2019. Gastrointestinal dysmotility is considered as one of the two main underlying pathophysiological mechanisms for development of functional pain. Major component of my previous studies concentrate on assessing gastric motility in children with this condition. Several studies I conducted on gastrointestinal motility in affected children have demonstrated abnormal gastric emptying and antral motility in all four main types of functional abdominal pain disorders, that is functional abdominal pain, irritable bowel syndrome, functional dyspepsia, and abdominal migraine. Furthermore, there was a significant correlation between motility abnormalities and symptoms, which highlighted the importance of gastrointestinal motility as an underlying pathophysiological mechanism. In addition, children with irritable bowel syndrome who were exposed to emotional stress had more severe impairment of gastric motility than those who are not exposed to such events, which highlighted the impact of psychological factors on gastrointestinal functions. Allergy and immune activation have been suggested as a possible pathophysiological mechanism for functional abdominal pain. Therefore, we evaluated the association between asthma and functional abdominal pain. In this study, we found a significant association between asthma and different types of functional abdominal pain. Finally, based on available information, we published two main models to describe the pathophysiology of functional abdominal pain in World Journal of Gastroenterology in 2018. The bottom-up model proposes that peripheral factors in the gut, such as low-grade inflammation, post-infectious changes, altered gut microbiota, immune activation, altered serotonin and bile salt metabolism, and increased intestinal permeability in combination or in association play a critical role in altering the pain perception and related behaviors. The other model, the top-bottom model, suggests that the main pathophysiological mechanisms initially involve in brain. In this model, the primary disease processes in the brain with the interact with peripheral organs through brain-gut axis to generate alterations in gut microbiome, immune functions, motility, secretion, and intestinal epithelium, which ultimately lead to clinical expression of functional abdominal pain. How can you manage functional abdominal pain? This is the fourth question we try to answer during my research work. Without thorough understanding of pathophysiology, it is not possible to devise a rational therapeutic strategy. Currently recommended management for functional abdominal pain include explanation, reassurance, and different supportive management therapies. Many therapeutic agents have been investigated to manage this condition, but so far there is limited evidence of their effectiveness. Domperidone is a recognized prokinetic agent and is known to improve gastric motility. Therefore, we conducted a double-blind placebo-controlled randomized trial to assess the efficacy of domperidone in management of this condition. The preliminary results have shown a significant improvement in antral motility as well as in symptoms following administration of domperidone at 8 weeks and 6 months. This is a brief highlight of my quest to find solutions for pediatric functional gastrointestinal diseases. Let's look at the key messages conveyed during my journey. Functional abdominal pain is a common health problem in children around the world and has a significant impact on their quality of life. Psychological factors such as emotional stress, exposure to abuse, personality abnormalities are important risk factors for this condition. Gastrointestinal dysmotility is an important underlying pathophysiological mechanism for functional abdominal pain. Prokinetic agent domperidone helps in the effective management of affected children. However, this is not the end of my quest, so the journey will continue. Main steps in my quest will be, in the future, I like to collaborate internationally to assess the global burden of pediatric functional gastrointestinal diseases, explore pathophysiological mechanisms further to describe exact underlying etiology of this condition, and also to conduct trials to identify pharmacological and non-pharmacological interventions and to formulate effective management guidelines for functional abdominal pain. Goal of my quest is to lessen the suffering of children with functional abdominal pain and keep their smile forever. There are many people who helped me in this quest. First and foremost, I'm indebted to Dr. Asoka Disanayaka, former professor of physiology, who encouraged me to pursue a research career in functional gastrointestinal disorders, and also my supervisors of Doctor of Medicine degree, Senior Professor Janaka De Silva and Emeritus Professor Harendra De Silva, and my PhD supervisor, Professor Mark Benninga of the University of Amsterdam, for their guidance and support throughout my research work. I was fortunate to have two dedicated PhD students, Professor Amaranath Karunanayaka and Dr. Manori Vijayakumari, who collaborated with me in some excellent research projects. Technicians of the Gastroenterology Research Laboratory, Janeshwari, Janaki, and Ratnayaka were very helpful when I was conducting motility studies. All the members of the Department of Physiology supported me in several ways when I was conducting my research work. My parents were dedicated and looked after my daughter, which allowed me to engage in research with peace of my mind. My daughter was very supportive in all my research activities, never complained, never disturbed. I'm very fortunate to have her as my daughter. And also, finally, my husband, Shaman, is also my co-researcher in all these research projects, worked hard with me, supported me, and encouraged me. He's the pillar behind all my success. Thank you very much.
Thank you, Madam. Ladies and gentlemen, now I cordially invite Professor Nilanthi De Silva, Vice Chancellor, University of Kelaniya, to grace the occasion.
Good evening. Can you hear me?
Yes, Madam.
Yes, yes, we can.
Okay, good. Good evening, ladies and gentlemen. Professor Niranga Devanarayana, President of the Physiological Society of Sri Lanka and members of a council. Vidyanidhi, Professor Mangala Gunathilaka, the orator for this evening. Members of the Physiological Society, distinguished invitees, ladies and gentlemen. Let me first thank the President and the Council of the Society for inviting me to be the chief guest this evening on the occasion of the inauguration of your 33rd annual sessions. I consider it a signal honor to speak a few words to you today, to a society with a very long and distinguished history and a membership of many eminent scholars. As the Vice Chancellor of the University of Kelaniya and a former Dean of the Faculty of Medicine, I'm very proud of the fact that our Professor of Physiology is the current President of your society. I remember Niranga well as a bright young probationary lecturer and have watched her grow and mature into an excellent researcher and a senior academic in her chosen specialty of gastrointestinal physiology. In a similar fashion, our medical faculty has also matured into a place with pretty good human and physical resources. I don't think I'm mistaken when I say that we now have come of age and we are able to pull our weight alongside much older medical schools. We have invested a great deal over the years in recruiting the best possible medical and healthcare professionals and nurturing them into experts in their chosen field, who are also at the same time competent teachers who enable our students to become good doctors. At the same time, we have created a research culture within the faculty that has enabled the University of Kelaniya to rise in the international rankings. And I know for a fact, as evidenced by what Niranga just presented actually, that she has contributed a great deal to that particular aspect. As a university, we have also invested a lot in developing our IT infrastructure, both at Dalugama and at Ragama. And that is why we are able to host this virtual conference for you. In fact, hosting virtual conferences seems to have become the new normal. This is the fourth online conference that the university has hosted just this week. The COVID-19 pandemic has made us change how we lead our lives on a day-to-day basis. Some of these changes, I think, will melt away as the pandemic recedes. But I hope that the use of virtual platforms to bring people together for learning opportunities will stay, because it reaches across so many boundaries. On the other hand, looking at the list of people participating in the meeting today, in today's inauguration, I felt it would have been quite nice to have met in person, because I see so many old friends in that list. And it would have been lovely to have an opportunity to talk to all of them this evening. To be honest, I found it quite difficult to figure out what I should talk about this evening. As you heard, as you may know, I'm a parasitologist, and parasitology has little to do with physiology. But when I think back to my time in medical school, I must say that I enjoyed learning physiology. In fact, it was the only subject for which I earned a distinction when I was in the Colombo Medical Faculty, not parasitology. And I asked myself this last few days, why was that so? I think part of the reason was because I had heard so many horror stories about how hard it was to pass the second MD examination those days. And like many other newcomers to the medical faculty, I studied extremely hard out of a simple fear of failing and having to take the exams again. However, that is only a small part of the story. I think the main reason was really because we had great teachers of physiology, people like Professor Carlo Fonseca and Professor Colvin Gunratna, who made the subject come alive. Of the three subjects that filled our preclinical years, for me, physiology was the one that introduced us to all the glorious complexities of how the human body functions. Physiology was the subject that enabled me to understand the basic principles and concepts in relation to each of our organ systems, and then use that understanding in real world situations. And if students don't get those basics right, the rest of the medical course becomes really difficult, doesn't it? And so somebody who has, as someone who has spent quite a bit of time and energy in medical education, in addition to parasitology, I must acknowledge the key role played by teachers of physiology in holding a medical curriculum together. You are the people who lay that all-important foundation for students to enable them to build on that basic understanding and learn other subjects that comprise the rest of the medical curriculum. You are the people who form that first contact with our medical students and inspire them to learn more. I'm sure you are well aware of the crucial responsibility that physiologists bear when it comes to undergraduate medical education. So I won't belabor the point, but I hope that you will keep it in mind, especially the younger teachers, the younger members of the society. I don't intend to speak at length, and so I will just end my few words by wishing all the members of the Physiological Society of Sri Lanka fruitful deliberations. I saw that you have a very interesting set of talks lined up for this evening as well as for tomorrow. And so I wish you a very successful annual sessions. Thank you.
Thank you very much, madam. Dear all, the next up on the agenda is the awarding ceremony. Here is the winners. Professor K. Senaviratne Memorial Research Award for 2021 goes to Dr. A.M. Manori Vijayakumari. Dr. Kumari, who is currently holding the post of Senior Lecturer, Department of Physiology, Faculty of Medicine and Allied Sciences, Rajarati University of Sri Lanka. She was a past medical student from the Faculty of Medicine, University of Kelaniya, with a second honor class, and obtained her PhD 2019 in University of Kelaniya with the title of The Association Between Asthma and Functional Abdominal Pain Disorders in Pediatric Population, Common Pathophysiological Mechanisms Causing this Association. She published her findings of PhD in 2 SCIE Index Journal, World Journal of Clinical Cases in 2021. She won 3 awards for her PhD findings. The Dean's Award in 2019 for her publication in SCIE Journal from the Faculty of Medicine and Allied Sciences, Rajarati University of Sri Lanka. The Best Poster Award for one of her abstracts published in 6th Global Congress for Consensus in Pediatrics, Colombo, Sri Lanka in 2017. And a travel award to participate and present one of her abstracts at the 14th Asian Pan-Pacific Society for Pediatric Gastroenterology, Hepatology and Nutrition in 2018. Second award, Professor K. Senaviratne Memorial Award for Physiology 2021 goes to Mr. J.P.O.R. Jayakodi. Mr. Jayakodi is a past student of Bandaranaike College, Gampaha. He is currently a third-year student at the Faculty of Medicine, University of Colombo. He obtained a first class for the Basic Sciences stream with distinctions in Anatomy, Biochemistry, and Molecular Biology and Physiology. And also he obtained the Here is the certificate for winners. Now I call upon Professor Niranga Devanarayana, President, Physiological Society of Sri Lanka, to give the introduction of the orator.
Late Professor K. Senaviratne is the second Sri Lankan professor of physiology and has contributed tremendously to uplift the field of physiology in Sri Lanka. In memory of this great physiologist, the Physiological Society of Sri Lanka conducts the prestigious K. Senaviratne Memorial Oration every year. Today, the K. Senaviratne Oration is delivered by Vidyanidhi Professor Mangala Gunatilaka of the Department of Physiology, Faculty of Medicine, University of Colombo. The title of the oration is Rabies Revisited, Present and the Way Forward. Is closely related to neurophysiology, which was the main research interest of late Professor K. Senaviratne. Vidyanidhi Professor Mangala Gunatilaka was born in June 1963. She had her school education from Vishaka Vidyalaya, Colombo. Afterwards, she entered the Faculty of Veterinary Medicine and Animal Science, University of Peradeniya, and graduated in 1991 with second-class honors and distinction in microbiology, physiology, parasitology, and public health. Subsequently, she joined the Department of Physiology, University of Colombo, and obtained her PhD from the same university. Professor Gunatilaka is a fellow of the Sri Lanka College of Veterinary Surgeons, and she is the president-elect of the college. Professor Gunatilaka completed her MSc in Laboratory Animal Sciences at Aachen University, Germany, 2020, and now she is a federation. Now she is in the Federation of European Laboratory Animal Science Association, recognized specialist in laboratory animal sciences. Professor Gunatilaka is the founder president of the Sri Lanka Association of Laboratory Animal Science, established in 2012. She has introduced laboratory animal science education to Sri Lanka in 2011 by conducting the first certificate course. She is the country representative in International Council of Laboratory Animal Science. During her tenure of office as the president of Laboratory Animal Science Association, She introduced several alternative models for animal experiments, and she founded the Society for Alternatives to Animal Testing in Sri Lanka. She has received several national and international awards and scholarships in recognition of her achievements and contributions. She is a recipient of many research and development grants from national and international organizations and supervised several postgraduate students. She has many publications to her credit in forms of conference papers, journals and newsletter articles, books and e-learning publications. In recognition of her outstanding contribution to science, Professor Mangala Gunatilaka was awarded the national honors Vidyanidhi by His Excellency the President of Sri Lanka in 2019. Professor Mangala has been an active member of PSSL for more than 2 decades. She was the president of the PSSL in 2010, in which I had the privilege of serving under her as the secretary. Furthermore, Professor Gunatilaka is the joint secretary of the South Asian Association of Physiologists since 2012 and a member of the Education Council of Federation of Asian Oceanic Physiological Societies. As the current General Secretary of South Asian Association of Physiologists, she is continuing her effort to improve physiology and research in the South Asian region. It is a great pleasure for me to invite Vidyanidhi Professor Mangala Gunatilaka to deliver the K.M. Senaviratne Memorial Oration for the year 2021. Over to you, Madam.
Good evening, everyone. President of the Physiological Society of Sri Lanka, Professor Devanarayana, and members of the Executive Committee, members of the family of late Professor K.M. Senaviratne, physiologist who joined from overseas, my dear teachers, colleagues, friends, students, ladies and gentlemen. It is indeed an honor, and I appreciate the invitation extended to me by the President and members of the Executive Committee of the Physiological Society of Sri Lanka to deliver the prestigious K.M. Senaviratne Memorial Oration 2021. Thank you, Professor Devanarayana, for your kind words of introduction. Professor Kirti Nisanka Senaviratne was born in Alpitia, Gaul on 22nd November 1929 as the second son of Dr. Robert Senaviratne and Mrs. Laura Senaviratne. He had his primary and secondary education at Royal College, Colombo, and during his school days, he won the Arunachalam Prize for General Knowledge in 2 consecutive years. Upon completion of his school education, he entered the Colombo Medical School in 1949 and obtained his MBBS degree with honors in 1954 from the University of Ceylon, Colombo, gaining a distinction in medicine and winning the gold medal for operative surgery. After completing his clinical training as a young doctor, he joined the Department of Physiology of the Colombo Medical School as a demonstrator in 1957. Subsequently, he obtained his PhD from the University of Edinburgh, UK. Professor Senaviratne had been promoted to the post of Professor of Physiology at the age of 39 in 1969 and held this post until 1981. Institute of Postgraduate Medicine, which later became the Postgraduate Institute of Medicine, was established by Professor K. Senaviratne in 1974. And he had worked there as the founder director until he was appointed as a regional advisor to the World Health Organization in 1981 and continued to work in the same capacity until 1986. His area of specialization in research was neurophysiology with special attention to diabetic neuropathy. Late Professor K. Senaviratne had been a keen researcher who identified the importance of animal-based research in medical discoveries. He used the isolated sciatic nerves of healthy and alloxan diabetic rats to determine permeability of blood nerve barriers in diabetes and understood why diabetic patients develop nerve damage. He had many research publications in accredited international journals to his credit. With the untimely death of Professor Kirti Nissanka Senaviratne on August 10, 1986 in Bali, Indonesia, Mother Lanka lost a brilliant scientist and a brave teacher. His brother, Mr. Nihal Senaviratne and family established a fund in 1999. With the support of friends and pupils of late Professor K. Senaviratne to award a prize in memory of this great physiologist. This prize is awarded as K. Senaviratne Memorial Gold Medal for Physiology to the student who obtains highest marks in the basic science stream examinations in physiology conducted by the Faculty of Medicine, University of Colombo. Furthermore, Mr. Nihal Senaviratne and family established another fund in 2010 for awarding Professor K. Senaviratne Memorial Award for Physiology, which is a cash prize of 50,000 rupees to the student who obtains highest marks at the physiology final examination of the basic science stream of Colombo Medical Faculty. Ladies and gentlemen, with that commemorative note about late Professor K. Senaviratne, I wish to focus on my oration. The title that I selected for the K. Senaviratne Memorial Oration 2021 is Rabies Revisited, Past, Present, and the Way Forward. This oration consists of 3 parts as indicated here. In the first part, I focus on information from a laboratory, pathogenesis of rabies, and global perspective in brief. Reporting from the Louis Pasteur's laboratory. Louis Pasteur was a French chemist and bacteriologist who became the founder of microbiology. At the inception, he had concentrated on studies related to bacteria and put forward his germ theory of diseases. His investigations had enabled him to develop the process of pasteurization and also to develop vaccinations for many germ-borne diseases. During these days, as rabies was also prevalent, he had extended his investigations into this deadly disease. He had inoculated the rabies virus into different animal models maintained in his laboratory. These investigations led Louis Pasteur to develop his rabies vaccine by growing virus in rabbits and subsequently drying the affected nerve tissue to weaken the virus. The report titled Rabies Vaccine saves boy and pasture indicates, I quote, on July 6, 1885, the vaccine was administered to Joseph Nestor, a 9-year-old boy who had been attacked by a rabid dog. The boy survived and avoided contracting rabies, which would have almost certainly proved fatal. Good thing it worked. Pasteur was not a licensed physician and could have been prosecuted had the vaccine failed. The legalities were forgotten, and Pasteur instead became a national hero. I close quote. As stated in the report, by the end of 1885, several more desperate rabies-exposed people had traveled to Pasteur's laboratory to be vaccinated. During 1886, Pasteur had treated 350 people with his rabies vaccine, of whom only one had developed rabies. Rabies is one of the oldest and most devastating zoonotic diseases known since 2300 BC. The word rabies originates from the Latin word rabere, which means anger and grave. Rabies is an acute encephalitis, which is an acute infection in the central nervous system caused by a rod or bullet-shaped single-stranded RNA virus, belongs to the Rhabdoviridae family. When a rabid animal bites or scratches a person, rabies virus is inoculated and get multiplied at the site of infection. Subsequently, virus binds to the nicotinic acetylcholine receptors at the neuromuscular junction and enters axons of the peripheral nervous system. Upon entering the spinal cord, virus travels to brain. Viral infection of brain neurons leads to their dysfunction, causing rabies, which is a deadly zoonotic disease. According to WHO 2018 precision paper, rabies is responsible for an estimated 59,000 human deaths and over 3.7 million disability-adjusted life years lost every year at global level. As per the 2016 report, jointly published by the Food and Agriculture Organization of the United Nations, World Health Organization, World Organization for Animal Health, and Global Alliance for Rabies Control, countries like Australia, Great Britain, Japan, and United States of America are free of rabies. Most of the rabies cases occur in Africa and Asia, with approximately 40% of cases in children aged less than 15 years. This deadly zoonotic disease is endemic to Sri Lanka. Rabies, being a major public health problem, imposes heavy economic burden too. Total estimated cost on the activities undertaken for its prevention and control is approximately US $8.6 billion per year at global level. Ladies and gentlemen, in the part 2, I am going to focus on dog vaccination and research-based evidence. All mammals are susceptible to rabies. The virus transmission happens via two cycles, namely urban and sylvatic cycles. Dog is the principal transmitter of virus in the urban cycle, while wild animals such as foxes, wolves, jackals, bats, raccoons, skunks, or mongoose are involved in the sylvatic cycle. Contribution of sylvatic cycle in rabies virus transmission is very low compared to the dog-mediated rabies, and the latter is responsible for up to 99% of human cases in rabies endemic countries, including Sri Lanka. Therefore, vaccination of dogs against rabies is identified as an important control measure to eliminate rabies. However, as per the recommendation of global organizations, vaccination coverage in dogs should be more than 70% in high-risk countries in order to establish and maintain herd immunity against rabies, and thereby to break the rabies transmission cycles. This shows the progress of dog anti-rabies vaccination and human post-exposure treatment, leading to reduction in number of human deaths over the last 40 years in the country. In this figure, purple bars indicate number of dog vaccinations, green bars indicate number of human anti-rabies vials used in post-exposure treatment, and orange line depicts the number of human rabies deaths in each year. Number of human deaths recorded in 2020 is 31. This figure also indicates that zero human rabies deaths could be an achievable target for our country. According to dog population surveys conducted as part of the PhD study by Dr. Rumini Piburage, the total dog population is approximately 3 million. Among them, approximately 30% is free roaming. This makes it little difficult in achieving WHO recommended dog vaccination coverage. Vaccination of dogs leads to development of immunity against rabies virus. There are several factors which affect immunity development following vaccination. Among the factors, the potency and efficacy of the vaccine are two important considerations. Potency is the ability to exert the desired effect in dogs following vaccination. Efficacy is a measure of lowering the risk of an identified disease. Efficacy of vaccination against a disease could be determined by the immune response against the disease. Humoral immunity measures the antibody response, which is B lymphocyte mediated response, and the cellular immunity or T lymphocyte mediated immune response is responsible for destroying foreign antigens. This figure depicts how humoral and cellular immune responses are initiated by a foreign antigen. For example, by the killed rabies virus antigen included in the anti-rabies vaccine. Humoral and cellular immunity act against the virus to control the disease if the animal is exposed to the virus. Also, during the initial or primary immune response, which occur In the animal following the first anti-rabies vaccination, memory B and T cells also develop, which are useful in boosting up an immune response in subsequent anti-rabies vaccinations. The presence of proportionately higher percentage of free-roaming dogs in the country is a challenge faced by the authorities in rabies control and elimination. During my clinical practice as a veterinarian, I encountered several occasions of vaccination failures, especially in puppies. On the other hand, there was no established mechanism to monitor anti-rabies vaccine efficacy routinely in the country. I pioneered in immunogenicity studies in dogs following anti-rabies vaccination in the country, in collaboration with the Department of Rabies Research and Vaccine Quality Control of Medical Research Institute in 1999. The first project design was to determine virus neutralizing antibody titers in dogs or humoral immune response before and after vaccination with anti-rabies vaccine. This project was funded by the National Science Foundation, and dogs brought from Port and suburb were recruited to the study. In this study, adult dogs aged 1 to 5 years and puppies of 3 months old were considered under 2 groups. Groups were subdivided considering previous vaccination history of adult dogs and the vaccination status of the female dogs of puppies recruited. In this study, only domestic dogs were included. Government Rabies Control Program recommended, World Health Organization approved imported vaccine was injected intramuscularly to animals after collecting day 0 blood sample, and post-vaccination blood samples were collected in 1 month, 6 months, and 1 year. Serum samples of blood collected from 52 animals were analyzed for rabies virus neutralizing antibody titers by rapid fluorescent focus inhibition test. At the time of commencing this project, the recommendation for the first anti-rabies vaccination to puppies was at the age of 3 months. The second project was planned as an extension of the first project, with the inclusion of apparently healthy stray and domestic dogs. This was conducted by Dr. Uvini Pimburage as the first part of her PhD. National Science Foundation was the funding organization. Local breed dogs from Kalutara district were recruited for the second project. Considering the age, previous anti-rabies vaccination history, and regularity of vaccination of juveniles and adult dogs, and the vaccination status of female dogs of puppies, recruited animals were subdivided into 8 groups. Serum samples of blood collected on identified time points from 380 animals were analyzed by the rapid fluorescent focus inhibition test for rabies virus neutralizing antibody titers. Mean antibody response or humoral immunity to anti-rabies vaccination in adult dogs and puppies recruited in Project 1 is included in these 4 charts. The antibody response provides evidence for the efficacy of the vaccine, as it has induced an immune response following vaccination. Dogs having a previous vaccination history shows a higher response compared to the dogs without previous vaccination history. The absence of maternal immunity in puppies of vaccinated female dogs is evident at the time of first vaccination. The antibody response considered as protective against rabies is 0.5 international units per milliliter. This chart shows the antibody response in adult dogs, juveniles, and puppies recruited to 8 subgroups in the Project 2. The pattern of immune response in adult dogs and puppies was closely similar to the response observed in Project 1. Based on the findings of 2 projects, our conclusions and recommendations were: maternal antibodies do not provide adequate protection to puppies until the first anti-rabies vaccination. Immunity development after immunization seems to be closely similar in both the groups of puppies, irrespective of maternal immunization. The first anti-rabies vaccination for puppies needs to be done before the age of 3 months, as protective antibody titers were absent at that age. Thereafter, a booster vaccine to be given at a suitable interval, as protective antibody titers were not maintained until 6 months in puppies. After the recommended 2 anti-rabies vaccinations to puppies and juveniles in the first year, annual boosters to be given to all dogs in order to maintain the antibody titers above the protective level, as rabies is endemic to Sri Lanka. Results of the first project with the recommendations made were submitted to Ministry of Health, as it was the responsible authority which organized government dog vaccination program. Results were published in Ceylon Journal of Medical Sciences. The 2 projects completed measured actual antibody titers following anti-rabies vaccination in dogs. These findings, as the only research-based evidence, were considered to revise dog anti-rabies vaccination schedule in the country in 2013. Results of Project 2 was published in an accredited international journal in 2017. Our contribution for the application and advancement of science was identified by the Science and Technology Ministry for a national award under the category of multidisciplinary team efforts in 2016. Upon completion of the second project, I received a recommendation from the funding organization to look for the effectiveness of the revised dog vaccination schedule. Therefore, the third project, titled Pattern of Immunogenicity in Domestic Puppies and Juvenile Dogs following primary and subsequent booster vaccination against rabies in Dehiwala municipality area, was planned and dogs were recruited, and samples were collected until 1 year following vaccination by a veterinary surgeon attached to Dehiwala municipality. These samples are being analyzed by rapid fluorescent focus inhibition test at the Medical Research Institute. Ladies and gentlemen, now I am going to focus on the last part of this oration. It is the way forward. The information I mentioned in part 2 are related to humoral immunity, measurement to determine the efficacy of vaccination. There is no established method in the country to determine cellular or cell-mediated immunity following anti-rabies vaccination. There are about 7 different brands of anti-rabies vaccines meant for animals marketed in the country as single-dose and multi-dose injection vials. Registration of the vaccines are based on information dossiers of these imported vaccines submitted to regulatory authorities by the local agent. Potency of these vaccines has not been tested in the country. Therefore, a new project was designed to compare immunogenicity and potency of these different brands of animal anti-rabies vaccines. In this type of a study, experimental conditions need to be standardized to minimize variation as much as possible. This new project includes an experimental study involving laboratory-bred rabbits. Thereafter, 3 vaccines of highest immunogenic capacity and good potency will be selected for the planned field study involving dogs. This is a multi-center collaborative study which includes Animal Center of Medical Research Institute, Colombo Municipal Council, Public Health Veterinary Services of Ministry of Health, and the International Collaborating Laboratory. Samples are analyzed by fluorescent antibody virus neutralization test at the WHO OIE reference laboratory for rabies and wildlife in France. Once completed, this will be the first project at global level where rabbit is used for immunogenicity studies following anti-rabies vaccination. At the same time, for this project, a semi-quantitative assay for the detection of antibodies, that is to check whether antibody titer is at the protective level or not, was established in the Department of Physiology, Faculty of Medicine, University of Colombo. Currently, the method used for anti-rabies vaccine potency testing is the National Institute of Health mouse inoculation test, where quite a high number of mice needs to be sacrificed. During this procedure, mice may develop rabies if the potency of the anti-rabies vaccine tested is not up to the required level. Our attempt is to see the possibility of using immune responses as a substitute for mouse inoculation test. Funding organization is Boehringer Ingelheim. I hope that the efforts we have taken will be useful to achieve the WHO recommended vaccination target in the country. In conclusion, among the control measures, dog vaccination plays a major role in preventing human rabies deaths in Sri Lanka. Team effort of relevant organizations is a must to achieve the recommended vaccination coverage, while adhering to revised vaccination schedule using anti-rabies vaccines of good immunogenic capacity. This not only develops herd immunity in the dog population, but also makes rabies elimination is an achievable target. Multi-stakeholder concept has a real impact on eliminating rabies from our beautiful motherland, while contributing to One Health approach in order to reach the global target of 0 deaths by 2030. Ladies and gentlemen, I could not have come to this stage without the support of my collaborators, including the research students in these research projects. Dr. Rubini Pimburage completed the first PhD project on rabies in the country. Dr. Hasanti Divakara is involved in the new project. 2 ladies in the collaborating teams need a special mention. Dr. Umala Vimalaratne, who was the head of Department of Rabies Research and Vaccine Quality Control at MRI when I planned my first study. The discussions I had with her during planning were very encouraging and positive. Dr. Florence Cliquot of France accepted me in her laboratory for a training and also agreed to collaborate to conduct the new project. As this kind of studies need high level of funding, she not only accepted to collaborate, but also recommended me to Dr. Jean-Christophe Thibault, who was a director at Boehringer Ingelheim, to fund the project. I highly appreciate the support of all collaborators. I thank all who supported me indirectly for the success of the projects. I am very thankful to my husband Kirti and 2 daughters, Dr. Shehani and Kavindya, for their encouragement and support at all times. Ladies and gentlemen, I thank you all. Thank you.
Thank you very much, Madam. We have now come to the end of the program. I invite Dr. Naomi Ranatunga, Secretary, Physiological Society of Sri Lanka, to give the word of thanks.
Good evening, everybody. Senior Professor Delanti de Silva, the Chief Guest, the Vice Chancellor of the University of Kelaniya, Vidyanidhi, Professor Mangala Gunatilaka, the orator of Kane Senaviratne Memorial Oration, and Professor Niranga Devanarayana, the President of Physiological Society of Sri Lanka, Ms. Nihal Senaviratne and the family members of Professor Kane Senaviratne, the award winners, all the invited guests, council members of Physiological Society and all the members of Physiological Society of Sri Lanka, ladies and gentlemen. It is an honor and privilege for me to deliver the vote of thanks at this annual event. First of all, I would like to extend my sincere gratitude to Senior Professor Nilanthi De Silva for being with us in this valuable time, with spending your valuable time with us as the chief guest. You augmented the value of this evening. Thank you very much, Madam. Dear Madam, your talk on rabies was an eye-opener for the future challenges. Vidyanidhi, Professor Mangala Gunatilaka, the orator of Kane Senaviratne Memorial Oration for the year 2021, Thank you very much, Madam, for your enlightening speech. Further, I would like to thank Dr. Asoka Disanayaka, the orator of the inaugural Kala Fonseca oration, and Professor Nikhil Thapa, the orator of the Valentine Basnaik oration, to be delivered tomorrow. As always, your collaboration with Physiological Society of Sri Lanka was invaluable for years. I would like to express my heartiest gratitude towards Mr. Senaviratne and family on behalf of Physiological Society of Sri Lanka. I would like to extend my gratitude towards all the resource persons of annual scientific sessions of Physiological Society 2021, especially the chairpersons of the symposia and free paper sessions, plenary speakers, speakers of the symposia. And your expertise were very helpful. Dear Madam, dear Sir, thank you very much for your support as the abstract reviewers, as well as the reviewers, as well as the guidance given to upgrading these papers for a good piece of scientific work. At the same time, I would like to extend my thanks to all the judges of pre-paper sessions, and it will be a challenging task to choose the best papers. Thank you very much for your time and dedication in this regard. Adding to that, I would like to thank especially the 3 evaluators of K.N. Senaviratne Memorial Research Award. Further, I would like to thank all the members of the council for enormous support and guidance given throughout this event. Especially the Professor Niranga Devanarayana, the president of the PSSL, and Dr. Lakmali Amarasiri, the editor of Physiological Society of Sri Lanka. Dear Madam, your efforts were really thankful, and as always, you are the center of art in the society. On behalf of the PSSL, I would like to extend my thanks to Dr. Dilipa Ediravira and technical supportive staff of the Computer Center Faculty of Medicine, University of Kelaniya, and Mr. Samira Alvis and his staff for the audiovisual unit. And I would like to thank the main sponsor of the sessions, the Leeson's Hospital, Ragama. At last, but not the least, I should mention all the academic and non-academic staff members of Department of Physiology, Faculty of Medicine, University of Kelaniya, and the Department of Physiology, Faculty of Medicine, Vajayanagar University of Sri Lanka. Thank you very much for the ground support given throughout this process. If not for your support, this will not be a successful event. Dear members of Physiological Society of Sri Lanka and all participants in online platform, thank you very much for your time and for being with us. Thank you very much.
Thank you, Madam. So, on behalf of PSSL, I would like to take this opportunity to thank all the chairpersons and invitees who made their effort to come to the session today. And we are cordially inviting all of you to join with the second day of the scientific session tomorrow, 9 a.m. onwards, to gather knowledge. Thank you very much.